All right, next up we have Bioxytran Inc. They are on the OTCQB, trading under this ticker symbol BIXT. The company is a clinical stage biotech with two revolutionary platform technologies. They have an oral glycovirology drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that neutralizes the COVID-19 virus and prevents it from entering cells. They also have another platform drug being developed to treat strokes. So due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. But before we get into the heart of the interview, Mike, nice to see you again. Mike Sheikh, I'd like you to address the elephant in the room about COVID, right? Everyone's still talking about it. Now, earlier this year, President Biden indicated the public health emergency would expire on May 11th, allowing all business operations to return to normal. So, for all intents and purposes, COVID is pretty much over. So maybe you can comment on how COVID affects what you're doing as a company. And welcome back.
Hey, thanks, Anna. I'm really excited to be here. Now, I got to agree with you that COVID looks like it's over, but really, why is that? The government really isn't testing anymore, and they believe we've entered this endemic phase. This is the I see no evil, I hear no evil, I speak no evil part of government. Now, infection numbers, they're coming down, but we're not out of the woods yet either. India is now on the watch list by the WHO. There's this new XBB.1.1.6 variant, and it's really creating a surge in India right now. This variant is highly transmissible, and the first reports are basically indicating that it's not more severe, but what we do know that it's going to drive sickness and it's going to drive hospitalization. Now, the question is that going to spread outside our borders? We don't know, but we kind of do know, you know? But now let's pretend we know that this strain exists. Let's assume that it's going to fade away. Let's be completely optimistic. So does that mean that our company is going to fade away too? That's what investors really want to know. Are we going to fade away because COVID fades away? The answer is simple. We are, this is the read my lips part. We are not a COVID company. We want to end viruses. That's plural of viruses. COVID, it's just a case study. It's one part of our pipeline. A couple of weeks ago, we did a presentation to a bunch of scientists, and they understood the platform potential of our galectin antagonists. And honestly, they were pretty excited with that platform, but they were even more excited with our other platform, the oxygen carrier. So let me say that COVID is simply an indication, a case study, and it's something that's on our way to approval of this platform technology that deals with a number of viruses that includes COVID.
Well, thank you for that clarification, Mike. Talk a little bit more about your platform technology that can deal with other viruses. There's so many out there. Realistically, how big do you think this is? I mean, are we talking a dozen viruses, hundreds? Can you give us a sense of scale or limitations?
Okay, in our last webinar, we had a slide that revealed the galectin signature. And it basically has a number of DNA and RNA viruses. And this universe of viruses is broken down into DNA or RNA viruses. But galectins, that's involved in all of them. And that's the key. So overall, we're looking at thousands of these, what are called zoonotic viruses. Those are viruses that can translate to humans. And 250 of them that we know of, at least, can translate to humans. Now, there's roughly 26 families of human viruses, and we've shown activity in 3 of these families. That just goes to show you our broad spectrum activity, right? So before this, you'll see maybe one within a family, you'd see a little bit of activity, but not outside. So this highlights a broad spectrum activity. So So what we did was we run some in vitro studies using Prolectin-M. We did it on COVID, we did it on influenza, and we did it on RSV. We announced results a couple weeks ago, but honestly, investors really weren't enthused with it until this independent article came out and basically beat them over the head and said, hey, you got something, this experimental drug out there that can treat upper respiratory tract infections. Pay attention.
Well, and that's one of the reasons why you wanted to come on the conference today, correct? To talk about the peer-reviewed data. So why is peer-reviewed this journal, so important?
In one of our past emerging growth conferences, I said a peer-reviewed journal article would represent a major value inflection point. Well, when you look at the stock price, it doesn't look like that happened quite yet. But from the scientific perspective, it's a different story. I'm pleased to say that we've almost reached the viral status when you consider one of the biotech influences out there in COVID-19 tweeted our journal article out to the scientific masses. This Chris Turnbull guy tweeted our journal article out there, and I think he's at like 600,000 cumulative hits, and the number's still climbing as we're speaking. So there's a lot of engagement on his tweets too. So this is part of the scientific process. And as a result, my Twitter account, it's doubled in the past month. So from a scientific perspective, there's a lot of interest brewing, and we're actually working on scheduling some interviews with other science-based influencers that you're going to see in the near future. I love emerging growth, but we got to expand, okay? But now the primary reason that I'm excited about this peer review is this unwritten rule that's out there. It's like a prerequisite to getting on airtime on major media outlets. They won't even let you in the door if it's not peer reviewed. So this opens up a whole new of awareness possibilities. And you understand what we're about. We're trying to end viral infection.
Well, I saw that a couple of weeks ago, you did a presentation to several scientists. It was titled Ending Viral Infection. But this presentation also had a section on long COVID. That's a pretty hot topic as well. You wanted to talk about this here. But before we get into your long COVID presentation and then follow up with questions, let's ask this big picture question. Do you really think we can end the death associated with viral infection this century as we ended the death associated with bacterial infections last century? Is your technology like an antibiotic for viruses?
Okay, so the question, do I think we can end death from viral infection this century? The answer is yes. We have the building blocks of a very powerful platform technology that can quickly and efficiently identify whether or not certain carbohydrate drugs are able to react with the spike proteins on the surface of the virus. This field of research is called glycovirology, and we're essentially the pioneers of that. We're very excited to prove out more viruses. The 3 that we have so far, that is just scratching the surface. Our entry inhibitors are like nothing like antibiotics in terms of their mechanism. See, antibiotics, they're designed to target the cell walls of bacteria. Our drugs are designed to neutralize the spikes of the virus that enable that cellular entry. But the analog between antibiotics and entry inhibitors from viruses, it is kind of appropriate in the sense that last century, infections, they were the dominant drivers of death. And then antibiotics arrived on the scene and kind of squash that and definitely improve the mortality. Right now, let's talk about the mortality rate. It's going up, and people are talking about this excess mortality after COVID. And I think a lot of it, I think it can be traced to all this huge number of viral infections that we have. So directly, indirectly, COVID is kind of still driving death higher. So what does that mean? COVID wreaks havoc on your immune system. It's hard for people to recover. And this is kind of like the perfect segue for me to talk about my long COVID. So I'm going to now start talking about my long COVID slide deck. So we're going to kind of switch over to that. So here we have forward-looking statements. And then let's talk about what long COVID is. It's COVID that just keeps hanging out. It's COVID that lingers. Clinically, they say, look, you're not returning to your normal state of health. It's not explained by alternative diagnosis, and it's not due to active viral infection, which means if you have a nasal PCR and you have COVID, it's not classified as long COVID. It's classified still as COVID. It's disabling America. This is a really big deal. We need to pay attention to it. Look at this. 1 in 5 people with COVID that get COVID have long COVID. Look at the number of cases, 65 to 100 million, the estimate ranges worldwide have long COVID. There's lost work days. The economic burden is in the trillions, $3.7 trillion. And then this statistic, 7.5% of the people, that's in the United States, of the population are reporting having long COVID. And your quality of life decreases. It's not a good situation. And then let's look at the symptoms. The common symptoms here on the left slide, you'll see brain fog, fatigue, loss of smell, difficulty breathing, joint pain, digestive issues. And then look at all the organs that it affects. You see there on the right, look at these organs: heart, lung, skin, reproduction, stomach, head. And then look at all the subsymptoms. I mean, how do you characterize this disease? That's the next slide, the symptomology of it. How is it driven? What they did is there's a study that mapped ACE2. ACE2 is the primary entry mechanism for SARS-CoV-2. And what you'll see is the virus, you'll see the little virus in the center, and you'll see how it propagates itself all over to all these other organs. So ACE2 basically facilitates the spread. And then if you look there in the little kind of footnotes, we've mapped it in 72 different tissue types. That's a lot. But where does it mainly go? Look, you got the fatigue, the shortness of breath. You see that on the right. Look at these big bars. This represents the percentage of patients that have that symptom. So fatigue's number 1, and shortness of breath is number 2, joint pain number 3. So, a lot of that's, right up here in the head, or in the brain rather. This is long COVID is a heterogeneous disease. That makes it a very big challenge for clinical trials. There's testing of long COVID. What sort of test do you use? There's the endpoints, which is the clinical trial side. What endpoints are you going to use? So here's the problem. We have no validated tests or diagnostics that are readily accepted. I mean, there are out there, and there's really no industry guidance on what these endpoints should be. And then, to make matters worse, you have this other thing called short long haulers and long haulers, so that we can't even decide on what the disease is. Now we're going to get into my theories on long COVID, and there's a bunch here: viral persistence, organ damage, and microclots. We're going to lump those two together, and then autoimmune response and dysbiosis. So I'm going to go through each of these, and then we're going to go through our peer review. So viral persistence, what is this? Check this out. There's evidence out there that COVID stays in our tissues up to 12 months after infection. Whoa. Paxlovid treatment lowers the incidence of long COVID. Now, why is that important? Why are we looking at that as evidence of viral persistence? Well, because Paxlovid attacks the virus. And what did it do? In a study, we saw that it lowered, so 9.43 was the incidence per 100 people, and it went down to 7.11 per incidence. So basically, a little over 2 people per 100,000, it lowered the incidence just by taking Paxlovid. So treating that viral persistence helped for long COVID patients. So that's what we saw. And what's happening is we believe that there's this ongoing replication or viral reservoir that's driving long COVID. Now, there's another theory by Bruce Patterson, And he said viral persistence, like, it could be triggered by the S1 subunit, and that's driving the pathology. Now, I'm going to explain that very, I'm going to use an analogy. Let's say you have a gun and it's going into the cell wall. So a bullet is going to go into the cell. That's basically the virus spilling its guts into the cell, right? It's trying to kill the cell. Once it fires, you have this ejection cartridge that comes off. That's the S1 subunit. That's what happens. So we're saying all these cartridges are kind of laying around in your cellular body or in your body, and it causes irritation, So if a machine gunner sitting in a bunch of machine gun shells, you're just going like, well, this is irritating, right? It's kind of the same theory there. So we believe Prolectin-M, or Prolectin product, is going to stop infection by it binding to either the S1 subunit or active viral infection. So that's why we believe we have something really good to treat long COVID. The next slide talks about tissue damage and microclots. Okay, so there are these damage-associated molecular, these are the DAMPs, right? Pattern danger molecules. And what they do is they increase inflammation and coagulation. That's what their job is. And then galectins are part of the fibrotic process. So what galectins do is that they're part of the fibrosis. Fibrosis cuts off the supply to the organs, and that fibrotic process makes it hypoxic. It helps increase inflammation and coagulation. So what we're talking about there is by stopping the galectin-3, we're helping in the tissue damage and the microclot area. Now, here is an example of some microclots here. The theory is the spike protein is going to promote, there's a little graphic there, and there's this, there's the chart where you see the cascade that leads to a clot. That's the normal cascade that leads to a clot. What we're saying in COVID is it circumvents it. So you see it goes to the left and down and across. It circumvents it, and the theory is the spike protein of COVID is going to basically promote this and cause the clots. So you end up getting this unwanted clotting and inflammation that leads to it. Now, the next one is autoimmunity. This is a concept that's kind of a little bit hard to understand, but when you get COVID, what happens is antibodies are made, you get immune response, and they're called antibodies, and these antibodies are made to respond to COVID. But what happens is some people have some antigens that are on the surface of their cells right now that are kind of similar to the spike protein. So what happens is when you create these antibodies for COVID, and what happens is these antibodies are not created exactly perfectly for COVID. And what they do is they end up going to your body and COVID at the same time. And then what happens is you eventually clear the virus, but you've created these antibodies that also want to attack you. That's the problem. That's auto antibodies, or that's the layman's definition of what's going on there. And then finally, let's go to dysbiosis. Dysbiosis is basically the microbiome in your gut. It keeps everything in balance. But what happens when it's out of balance, you create a very, very favorable environment for these viruses to thrive, like Epstein-Barr virus. And Epstein-Barr virus is very much associated with that tired, fatigued, sleepy type of feeling. So what are we doing here for our galectin antagonist? We don't know if it works on Epstein-Barr or not. It may or may not, but we're saying there's the broad spectrum activity is very promising. And then finally, let me get into my development pipeline here. As you can see, we're going to go Prolectin-I. What we're going to do there is we're going to go in healthy patients in a phase 1. And the beauty about that is, and this is in India, the beauty about that is when we complete that, if the safety is there, wow, we can go into almost any phase 2 indication we want. Long COVID, some of these other ones, idiopathic pulmonary fibrosis, we could do massive label expansion. That's what gets me excited about that. Now, I said I was going to go over my peer review data, and that's what part of this was. So here, what we have is we have the control group and we have the active arm that was treated with our drug. Now, there's a red line there. It's sitting at 29, and we want to be above that line. I know it might be not make intuitive sense, but we're measuring CT values as the inverse of viral load. But we want to be above that line. Look at how quickly the treated group in green gets above that line, like almost like, bam, it's there. And then you look how everyone went to a point and stayed above the line. So that's a treated group, and look at the control group. Like one guy got to that above the line in 7 days. That's the night and day difference between the control group and our group. Now, here's another example of the results. This is on day 3. We had 82% were PCR negative, and then day 7, 100% were PCR negative. You see that at the top. We had 34 patients in the study, and my slide is okay. And then the other thing that we had there was on the right-hand side, you'll say we eliminated most symptoms by day 7. Now, why is that important for the long COVID people that are listening? Because when you get rid of the virus so quickly, you don't get into that stage that develops into long COVID with all these symptoms. So you see, we have barely any symptomology. We had like one guy with or gal with a cough, and that was pretty much it. Whereas where you have the orange ones there, a lot of the disease symptoms continue to persist in the control group. So we virtually eliminated it, and that's what was so telling about our studies, is not only do we have the biomarker proof, but we have the clinical data proof here. And that's pretty much it for what I wanted to talk about on the long COVID side of things. So back to you, Anna, for your questions.
Thank you, Mike. Can you also give us an update on the clinical trials and what some of your endpoints are? How long after starting the trial will it take to read out top-line results and then obtain approval?
Sure. In India, the first clinical trial is in the first clinical trial that we're doing. It's a dose optimization trial. Now, this trial is for 40 people. We're dosing 4 chewable tablets daily, up to 12 daily during the waking hours in the control group as well. We're measuring the percentage of people that turn PCR negative at days 3, 5, and 7. And we're also doing it, obviously, with the corresponding dose. Now, after this trial reads out, we're going to move on to do the equivalent of a pivotal phase 3 here, but over in India on 408 patients. And that's our PROTECT trial. That's what we call it, PROTECT. The results from our dose optimization are going to basically feed into that trial and determine the dosing. And then when we do it, we're planning on measuring PCR negative on days 1, 2, 3, 5, 7, 9, and 14. Okay, and all this is on clinicaltrials.gov. We're also planning on adding a follow-up in 30 days to tally like the number of people that still have these COVID-like symptoms. Now, this is a result, and this is my favorite part of the study, because the peer-reviewed journal article that I just went over and just talked about, you saw how we reduced the symptoms. We didn't have any rebound, so we're pretty optimistic that we're going to be able to eliminate COVID within that 30-day window in this upcoming trial. So we're excited about that. Now, with respect to the timing of the data readout, everyone wants to know about that. It honestly depends on how quickly we can recruit patients. If we're lucky and we get through the dose optimization quickly and raise enough money to start the phase 3 trial, we could be sitting in a really good position to complete this trial this year with a top-line readout. So we're very excited about that.
Fantastic. Mike, can you explain how the Indian regulatory process differs from here in the U.S. and our regulatory environment?
You know, some people might not realize, but the Indian regulatory climate is actually more restrictive than the U.S. counterpart. The Indian FDA equivalent is called the CDSCO, and their directive with respect to clinical trials. Their mentality is different. In the USA, we tell the FDA, here's what we're planning to do, and the FDA says, well, it's safe or it's not safe. The CDSCO directs what the endpoints of the study are going to be, as well as a trial design. It's also very risky for drug developers to do a clinical trial in India because of all the manufacturing control that you need to do in advance. So think about it. A drug company has to invest all this money to do Indian manufacturing, and you don't even know if the drug works. Now, this is a complete contrast to the US regulatory environment. So there's a huge upfront cost in India, and you cannot recover it. But there's an upside to this, and the upside is a swift and predictable approval process if the drug meets the endpoint. So many biotech investors might have remembered what happened with Viru Pharmaceuticals. They beat their endpoint, but then they got struck down by the US FDA AdCom advisory panel, and it was basically lack of robust safety data. In India, you don't get outcomes like this. Those things are unthinkable. The regulator designs a trial. They tell you how many people you need. They tell you the dosing. They tell you the endpoints. So if you meet them, they give you approval. Now, if you don't meet the endpoint, you don't get approval. So let's look at it from Bioxytran's perspective. We're developing Prolectin there. It's not risky for us. It's not risky at all. Look at our clinical trial results. The drug manufacturing elements completed. We have the approval to proceed. We also know based on statistics, our drug is highly likely to meet the endpoints. So the only thing left for us to do is execute. And for investors, they need to understand that's the risk.
Well, what do you make of the party's bioscience news this week? There's so many failed COVID drugs. It feels like Bioxytran might be the only one left standing. So what makes you think you will succeed when so many have failed?
Now, regarding the party's news, for those that might not know, they failed to meet their 3-day endpoint in standard risk patients with mild to moderate COVID. You kind of know where we stand on that. We got 82% eliminate, PCR negative on day 3. They were using a 3-CL protease very much like Paxlovid. So now here they are looking at strategic options. Now, what's interesting about this scenario is that Paxlovid withdrew from this type of study in early stage patients because it was too hard to show a difference between standard risk patients and because they got better so quick on their own. Now, if I had to speculate, the power of the study was probably off, or they didn't enroll people immediately after they tested positive. So it's a difficult thing to measure. Now, we're different. We think we have a really good clinical trial design because we're enrolling people that are quite sick. These people have CT values under 25, that's sick, and they enter the trial immediately. This gives us ability to really track the viral load as it goes down. Now, the reason we succeeded in our last trial with 100% responders rate is because we took a different approach in treating COVID. See, everyone else out there, including Pardes, is relying on the immune system to clear the virus faster for the person. But we have a novel approach of neutralization of the virus on contact. Our drug neuters these spike proteins' ability to infect cells. And then the liver eventually filters out the drug with the virus in tow. Our process of viral clearance isn't reliant on that immune system, which can be impaired when a person gets COVID. It kind of makes sense. So failure for us really isn't an option because we've already succeeded. Look at our clinical trial results. This is me pounding my fists on the table. Look at them. Look at the peer-reviewed report. Look at our p-value, 0.001, which means these results, they're not due to chance. We simply need to do another clinical trial. on a larger set of people to meet the regulatory requirements. Investors understand the risk.
Mike, we are out of time, but I want to congratulate you on all of this progress. This is really fascinating, life-changing technology that you are bringing to us. So I always look forward to speaking with you and hearing and learning so much. So please join us again in the future for some updates. We've got so many questions for you, but we've got to move on to our next presentation. So we'll send you the questions so you can answer on your own time. But thank you.
Thank you so much. Bye-bye.
Okay, everyone, stay with us. We'll be right back.